Greetings, everyone. Let's talk about the structure of DNA. That funny toy model you see in the background of this picture with James Watson and Francis Crick was the model that they built from what are essentially toys, molecular modeling, little plastic tubes and connectors. And it was that insight to get their hands and to get a concrete model to work with that was critical for them to be the ones to discover the correct structure for DNA. So let's dig in, and we're going to spend a few minutes reviewing some important scientific research that was instrumental in determining that DNA was the genetic and hereditary material of organisms. And I just want to warn you that I'll go quickly through this material, so take advantage of the format, the video lecture, and pause when you need to read the information that's on the slides. And please remember that I am not going to ask you to regurgitate any of the names or dates of this research. It's really just to give some background and context for how we got to understanding the structure of DNA. And I think it's important for that matter to know that there's a story with a bunch of people working on this and lots of folks contributing. All right, so well into the 20th century, we knew that organisms reproduced and that offspring resembled parents and that siblings resembled siblings. So this was things that we've known forever. And so there was an assumption that there was some information, this genetic information passed from parents to offspring, but we didn't know what it was. The leading candidates, because of what was found in the nuclei of all cells, the leading candidates were that the genetic information was being passed on by proteins or by DNA, because the nucleus is filled about 50-50 with proteins and DNA. And for a long time, proteins were thought to have been the best candidates for the genetic material because proteins are much more complex than DNA. But as we will find out, that assumption was wrong. So we're going to look at first some experiments by Griffith, who was trying to determine what was going on with bacteria that could cause a disease in animals, specifically bacteria that cause pneumonia, a lung disease in laboratory mice. And what was it about certain types of bacteria that could cause a disease when other types of bacteria did not make the mice sick? So Griffith knew that there were two similar strains of a particular species of bacterium, and that those strains could be grown on culture dishes, on petri dishes, and that one strain grew into colonies on the culture plate that were smooth around the edges, and that those bacteria were the ones that would make mice sick and kill them. And similar bacteria would grow in culture to form colonies on the petri dishes that were smooth and rounded, were rough around the edges. And those ones that had rough boundaries on the colonies were essentially harmless to the mice. The mice could be infected with those bacteria and they wouldn't get sick. So here are the four experiments that Griffith set up. First one, mice were injected with the disease-causing bacteria, and not surprisingly, they got sick and they died. Sorry, Mr. Mouse. Experiment 2, mice were injected with the harmless types of bacteria that grew in the colonies on the petri dish that had the rough edges rather than the smooth edges. Those mice after receiving the injections, would not get sick and would live. So he boiled the culture broth that contained the bacteria and therefore killed all the bacteria that were inside. Then injected some of those dead bacteria into the mice, and not that surprising, the mice lived. If the bacteria were killed, the assumption was that the substance may have been destroyed when the bacteria were killed, whatever it was that was the factor causing the mice to get sick. And then the one that was hard to figure. Griffith took some of the boiled bacteria, mixed them in with some living bacteria, but from the harmless variety. Those cultures that grew in the rough-edged colonies that had not made the mice sick in the past. He mixed some of the dead, killed bacteria that were the ones that made the mice sick with the living bacteria that didn't make them sick. And the mice that received both of those got sick and died. And when they did autopsies of the mice and collected bacteria from their lungs, the bacteria grew into the smooth colonies that were similar to the ones in the disease-causing bacteria to begin with. But those disease-causing bacteria that were injected into those mice were not alive. So how was it that this disease-causing factor in the dead bacteria was able to be transmitted to the living bacteria that were in the harmless bacteria? How was that transmitted and was able to change those harmless bacteria into deadly ones? Well, we know that this process is called transformation. And if you want to know exactly how it happens, I can tell you. But we're not going to do that in this video. It would be a little longer than I want it to be. But that transformation caused those harmless bacteria to become deadly. Something in the disease-causing bacteria was transmitted into the harmless one, causing them to change into the deadly form. So what was that chemical factor? And if that factor could change bacteria from harmless to deadly, maybe that was the factor that was also responsible for transmitting genetic information from parents to offspring. Well, another scientist, Oswald Avery, and the grad students and colleagues that Mr. Avery worked with repeated the same work that Griffith did and went further in trying to determine what was the factor that was able to cause those bacteria to be transformed. And so he took those boiled disease-causing bacteria and treated them with different enzymes that were able to chop up, basically selectively, all the kinds of organic molecules that you might find inside of a living cell. So he treated with enzymes, those extracts with enzymes that would chop up proteins. And he treated them with enzymes that would chop up lipids, ones that would chop up carbohydrates, and ones that would chop up nucleic acids. And then he used separate enzymes to chop up only RNA, which is one kind of nucleic acid, and to chop up only DNA, the other important type of nucleic acid. And when he did all of those experiments, he found that none of those other types of organic molecules was important, that he could destroy all of those types of molecules, proteins, lipids, carbohydrates, and RNA, and transformation would still occur. There was still something in the heat-killed bacteria that was causing the harmless bacteria to become deadly. And then he found out what that was by doing one more experiment in which he used an enzyme that would break down DNA, that would digest DNA. And when he did that to the extracts from the heat-killed deadly bacteria, the mice lived. So the transforming factor, the thing that caused the bacteria to change from being harmless to deadly, was DNA. And so Avery and his colleagues thought that maybe DNA is also the genetic material that's responsible for transmitting characteristics from parents to offspring. So a quick introduction to the structure of DNA, and then we'll talk more about this as we talk about how DNA functions in cells. Some basic facts. DNA is one of the 4 classes of organic molecules. It is a nucleic acid. DNA, like lots of other organic molecules we've been introduced to, is a polymer. A polymer made up of monomers. And the monomers, the building blocks of DNA, are called nucleotides. And all the nucleotides that are used to build DNA polymers are made of the same 3 ingredients. There is a 5-carbon ringed sugar that is deoxyribose. That's where the D in DNA comes from. There's a phosphate group, and there is a nitrogenous base. And here's what those nucleotides look like, and I've color-coded on this diagram the nitrogenous bases to highlight the differences between each nucleotide. So the nucleotides are categorized into 2 groups depending on if there's a double ring or a single ring structure. The double ringed bases are called purines, and the single ringed bases are called pyrimidines. You don't need to know that, but there's those words. And a single nucleotide consists of a phosphate, the 5-carbon ringed sugar structure, the deoxyribose, and the nitrogenous base, which is highlighted in the 4 different colors here. So I'm trying to outline right here a single nucleotide. And then adjacent to that, here's another nucleotide that is guanine for the base. And so there are 4 different building blocks, 4 different monomers that can make up DNA. And the order that those bases are lined up does not have to be AGCT, adenine, guanine, cytosine, thymine. It can be in any order that is on a single chain of the DNA. The sugars and the phosphates for all the nucleotides are exactly the same. And so this part of DNA is consistent up and down a long molecule of DNA polymer. And what's different from the individual nucleotides is the nitrogenous bases. And so a key piece of information that Watson and Crick had to consider was some information that was reported by a scientist named Erwin Chargaff. And he looked at lots of different species of organisms and determined that the ratio of guanine and cytosine in any species is essentially equal. So however much guanine was in a species' DNA, there would be essentially the same amount of cytosine. Likewise, however much adenine there was in a particular organism, there would be an equal amount of thymine. And that would be the same for any cell of any organism of the same species. And between species, the amounts of AT or GC would not necessarily be the same at all. Those could be quite different. But within a species, the amount of G and C would be the same, the amount of A and T would be the same. That was an important clue for Watson and Crick, as was the X-ray diffraction evidence that was created by a scientist named Rosalind Franklin. And she and graduate students in her group were taking pictures of X-ray diffraction. This was using X-rays to scatter in samples of purified DNA. And I know it doesn't necessarily show that from the picture here, but Watson and Crick and other people who knew a lot about DNA looked at that picture, and it suggested a structure to them. It suggested that DNA was a double helix. And we'll see what that means in just a second. So a double helix is like a twisted rope ladder. If you've ever seen like a rope ladder thrown over the side of a boat or a side of a building as like a fire escape, a rope ladder has the two sides made of rope and the wooden rungs in between. And so you can twist that rope ladder, and this is the way DNA is structured. And Watson and Crick were able to determine that by looking at the models that they were building and by looking at the X-ray diffraction pictures that Rosalind Franklin had taken. And they knew that A is the same as T and G is the same as C ratios that Chargaff had reported matched up with what they were seeing when they built their models. And we'll look at that in a second too. And here's exactly what's going on with that. So if we take a nucleotide, in this case adenine, and we take another nucleotide, thymine, and we orient them in opposite directions. So notice that the adenine has the phosphate pointing sort of up, and the thymine has its phosphate pointing down. If they're oriented in that manner, and we look at the atoms that make up adenine and thymine, they are able to form hydrogen bonds with each other the same way that water molecules do. And we know that individually a hydrogen bond is not a super strong attraction, but it's strong enough to give water some pretty crazy properties. And so if these two nucleotides were able to come in this orientation with each other, they would sort of stick together. Likewise, if the cytosine and the guanines were next to each other, guanine over here on the right, cytosine on the left, it was possible that atoms on either of those nucleotides could form 3 hydrogen bonds because the distances between those atoms was just exactly right for hydrogen bonding to take place. And there would be a kind of positive atom on one side, a kind of negative atom on the opposite side, straight down the row. And this was the clincher for Watson and Crick. They said, well, if we have a chain of nucleotides that goes in one direction, and if we have a chain of nucleotides that goes in the opposite direction, well then adenine and thymine have to match up because they would bond to each other in the middle here. Guanine and cytosine would match up because then they would bond to each other in the middle here. And that would create this stable connection between strands of DNA, single strands of DNA that are running in opposite direction. And that would create this double-stranded DNA if we look at both sides. That's a lot. I'll be happy to hear the questions you have when we see each other next.